研发术啊，研发术，他说同学你可是，你可是啊，你可是，没有是吧？我说我是谢振豪，公司，那个老师什么事？请坐，老师请坐。
我先把声音关掉，我先把。 我 live 了，然后我们现在去开外面连接。好。
除了那你毕业刚才当兵嘛？大家也是方面的。对。来，最好让你打一点好。对啊，现在海洋相关的高考还没有正式公布，但是就会现在在规划。很好啊，那里面有。
然后就建立个合作的。
我其实还算像我跟机械系老师欢迎如果有机会的话。我是说是生命讲其他，然后对，就是有认识吗？哎，不认识，对，自己跟你讲系的话，一样。大家都在这个场，常来的。对。 on the month of Friday, we have an environmental seminar and we'll provide it too. So the schedule already announced. You can check the schedule out on the website. So we have very diverse research topics to talk about. This is kind of an informal casual style of seminar. So during the seminar, you are feel free to continue to get more food or even after the seminar. Okay. So today it's our pleasure to have Professor Ming-Yang, and he's in the Department of Life Science, and he's an expert of photosynthetic biology and ecology. I will give the time to Ming-Yang to introduce himself. Let's give him a warm welcome.
Okay. Can everyone hear me? It's okay? Okay, so I don't use microphone. Thanks, Dr. Xie, for the introductions. So yeah, it's my pleasure to come here to give one an overview of what I'm doing at Department of Life Science. And just Dr. Xie said this seminar we have very diverse audience. So I decided we don't go too deep into the science at the beginning. Just looking at these topics, you feel it's very difficult to understand what's that about. So I decided we just talk about cyanobacteria first. I guess everyone heard about cyanobacteria. And then we still have time, we can go into deep into what I'm doing about them. So here's just some pictures showing cyanobacteria. They are quite diverse, have different morphologies and different sizes. So first, want to get everyone's impressions. So here you can see these are all bacteria. They have different colors, but not all of them are cyanobacteria. Can everyone make a guess? How many of them are cyanobacteria? How many of them are cyanobacteria?
All of them.
Oh, that's a good guess. That's a good guess. How about 1, 2, 2? Oh, one person. 3, 3. Okay. 4, 5. Okay. 6. All of them are cyanobacteria. So, okay, here is the answer. Only 5 of them are cyanobacteria. A, B, C, D, and F. A, B, C, D, F. So, although they are all colored bacteria, some of them do photosynthesis as well, but not all of them are cyanobacteria. And not all cyanobacteria are green, and not all green bacteria are cyanobacteria. So you can look at this A, looks like brown, very brownish, but it's cyanobacteria. And this one looks yellowish, it's also cyanobacteria. This is a Pochorococcus. I guess people working on marine biology knows about this. It's very abundant in marine environments. And this looks like a typical cyanobacteria color. It's bluish green. And for the other ones you see here, they look greenish, but they are not cyanobacteria. There are other kind of bacteria that do photosynthesis, such as green sulfur bacteria or green non-sulfur bacteria. Okay, so there are different colors of bacteria, and why we are interested in cyanobacteria. Because cyanobacteria is the only cyanobacteria that can do photosynthesis and produce oxygen. So in the talk today, I will briefly introduce you history of cyanobacteria and their morphology, the relationship between cyanobacteria and the plants, and where can you find cyanobacteria in our daily life. It's kind of relevant to what, where we live and our environments. And if we still have time, I would like to go into my research using flower light for photosynthesis in some cyanobacteria. But first, let's get some introduction about cyanobacteria. Okay, so the first thing you need to know is cyanobacteria are bacteria. So it's very easy. You can look at this scientific, its common name, cyanobacteria, so you can tell it's a bacteria. But some people, back to my high school, we called it as blue-green algae, 蓝绿藻. But because algae, it's eukaryotes, and cyanobacteria, they are prokaryotes. So actually, you cannot say cyanobacteria are blue-green algae because they are not belong to eukaryotes. But when back to several hundreds years ago, several decades ago, people don't know, when we don't have a good resolution of a microscope, it's very hard to differentiate cyanobacteria from algae just by looking at the morphology. They look pretty similar on the microscope. Until that time, we have a good resolution of a microscope, and we start seeing that these cyanobacteria, they don't have nucleus, and they are not eukaryotes. So we should not call it algae anymore. So in 1962, R.Y. Stanner and C.B. Van Neel, they suggest that blue-green algae must be classified under bacteria. And then this term became officially accepted by the society. And several more years, in photosynthesis-related publications, you don't see this term anymore. You don't see that anymore. And why cyanobacteria is important? You can see their relationship is distantly related to algae and plants. It's bacteria and eukarya. But because of plastic endosymbiosis, and cyanobacteria becomes the chloroplast of plant and algae. So this is the only lineage in bacteria that can produce oxygens, the same as in algae and plants. That's why cyanobacteria is so important in a bacteria. No other bacteria can do photosynthesis and produce oxygen like cyanobacteria and the algae do. So let's go back to here. Why some people still call it blue-green algae? Why they still call it 蓝绿藻? Okay, so I did a search on Google, and when I type 蓝绿藻, and probably you can also do that. When you type 蓝绿藻, you found, okay, what do you find? 蓝绿藻锭, blue-green algae, and this is a biotech company. And it's a PC home shopping website. And it's also 蓝绿藻, 螺旋藻锭. So what you find is nutritional supplement, 健康食品, about 蓝绿藻. And what do you find about 蓝绿菌? You find more about the scientific informations and classifications of these type of organisms. Like in Pensai 泛科学, and this is a kind of introductions of the 蓝绿菌. Also, you also find the information from the Wikipedia. So kind of differentiation between the scientific communities and common sense for the publics. They still don't use the term 蓝绿菌 too much. How about if you search the English term? When you search blue-green algae, what do you find? You always find a Wikipedia page about cyanobacteria here. But what you find here is not nutritional supplements. It's about how cyanobacteria, how blue-green algae affect our human livings. It's interesting, you see here, it's blue-green algae harmful to humans. And can blue-green algae make you sick? That's very strange to me. And how does blue-green algae kill dogs? How does blue-green algae kill dogs? Okay, I won't tell you the answer. Maybe you can Google that and find out that later. And if you search for cyanobacteria, you'll find pretty similar things. What does cyanobacteria do to humans? What does cyanobacteria do to dogs? So what does cyanobacteria do to dogs? Okay. Okay. Hopefully, after our talk today, you will find out what does cyanobacteria do to dogs. Okay. So there's another term for the nutritional supplements of cyanobacteria that they call it spirulina. The Chinese name is 螺旋藻. It has been used as a healthy nutritional supplements for years, and people can eat that. They put they made it as a powders or tablets and eat that as a medical pills. Okay, so looking at this term cyanobacteria and search from our Ministry of Education's website, you'll find out cyanobacteria there are several names, several Chinese names, 蓝菌, 蓝绿菌, and 蓝绿藻. I guess they are all acceptable based on our Chinese dictionary or translations. But I would prefer to call it 蓝绿菌because it's actually a bacteria but not algae. So let's go to some history about cyanobacteria. So I guess maybe back in your high school, you may see the picture like this. It's a stromatolites. It's a fossil. People call it as a fossil fields of. It's a fossil of cyanobacteria, and it can go back to 3.5 billion years ago. But it's not very conclusive that we can say cyanobacteria exists at that moment. The stromatolite is not a direct fossil records for the cell themselves. It's when the times we can see the cell clearly in the fossils, it's around 2.1 billion years ago. So it's still on debates whether cyanobacteria happen that early in the history of life, but at least we can find fossils around 2.1 billion years. And the occurrence of cyanobacteria back to that time is very important to the history of the Earth and the evolution of life. Why? Because it produces oxygens. So if you go back to the very early stage of the life starts with the prokaryotes, and oxygenic photosynthesis starts earlier than the oxygenic photosynthesis. So based on this hypothesis, cyanobacteria won't be the first prokaryotes that appears on the Earth. There must be some other bacteria got evolved earlier than cyanobacteria. So in the early stage of the Earth, there's no oxygens. Oxygen level is very low until the occurrence of the cyanobacteria start producing oxygens and then change the composition of the atmosphere, and then you can start seeing the aerobic eukaryotes using oxygen for respirations, and then endosymbiosis, algae, and then plants. That keep changing the level of oxygens on the Earth. Till now, it's around 20% oxygens. So I guess this kind of related to the topics of this seminar. It's about global climate change, and if you go back to history, the first climate change was done by cyanobacteria because they changed the compositions of the air. And what happened after cyanobacteria changed the composition of the atmosphere? Start producing oxygens, and what happens? So normally, those bacteria, they originally growing on the Earth. They don't like oxygens, and oxygen is very toxic to land. So these cyanobacteria keep producing these oxygens like pollutions to the other other members on the Earth. So they are basically killing all of the other bacteria at that time. It's oxygens that make the first extinctions on the Earth done by cyanobacteria. And they also kill themselves. But you know that life finds the way out. So the occurrence of the aerobics, eukaryotes, and the aerobic bacteria. So, and then because of the oxygens supports the respiration, so nowadays there's such a great diversity of the life. That because of the cyanobacteria at that times evolved photosynthesis and produced oxygens that changing the atmosphere. So, okay. So I keep talking about photosynthesis, but what is photosynthesis? If you look at this term, you can tell it's a synthesis using light, photosynthesis. So that's the chemical reactions you learn probably back in high school or in the universities. The carbon dioxide and the water under light to catalyze that and produce carbohydrates and oxygens. So in plants, that happens in the mesophyll of leaf. And there are two reactions called light reactions and carbon fixation reactions. So it's not called dark reactions anymore. It's called carbon fixation reactions. So cyanobacteria can do photosynthesis. And cyanobacteria is a phylum. It's a phylum, so it's a very large classifications. They contain many, many different cyanobacteria in that. And based on the morphologies, we can divide them into 5 types. So they are unicellular cyanobacteria. And based on their life cycles, we can divide it into 2. And they're also filamentous cyanobacteria, like a chain of cells. Some of them don't produce heterocyst, and some of them produce heterocyst and branches. So heterocyst is a specialized cell that can allow cyanobacteria to fix nitrogens from the atmosphere. So for this kind of cyanobacteria, they fix both carbon dioxide and nitrogen from the atmosphere. They largely can feed from the atmosphere by themselves. So how can the cyanobacteria do photosynthesis? So I guess most of you know the system in plants, and that system is quite similar in cyanobacteria too. So you can look at this cell of cyanobacteria. It pretty much looks like an individual chloroplast, ELUT, and it has thylakoid membranes inside the cell. On the membrane, you see those photosynthetic complexes, such as photosystem 2, photosystem 1. They harvest light energies and use the light energy to make the electrons jump from the ground state to the excited state, and following the electron transport chains, and finally used to produce NADPH and then ATP. That can be used for the carbon fixation cycle, carbon-Benson cycle. The thing's difference from cyanobacteria and plants is this antenna complex, phycobility zones. You only found this in cyanobacteria and some red algae. So this antenna complex can help cyanobacteria to harvest light energies and transfer the energy to photosystem 2 and photosystem 1. So they can harvest more light energy used for photosynthesis. So what's the benefit for having this huge complex? You can see as thin sections of the cells, this is thylakoid membranes, layer of membranes, and you can see this electron-dense particle sitting on the thylakoid membranes. They are phycobility zones, and seeing this, you can see in this cartoon figure. So they have peripheral rods and the central core. We call it allophycocyanins, 异藻蓝蛋白, and the rods could be either of these, phycoerythrin, phycoerythocyanins, or phycocyanins. So the allophycocyanin must connect with the phycocyanins, and the phycocyanins can connect to either PEC or PE. So you can see those proteins, they harvest different wavelengths of light. From the short wavelengths to the long wavelengths, there's a sequence. So because the short wavelengths of light contains a higher energy, the energy transfers from high to low level. So when the energy got harvested, the higher energy level at the PE, it can be transferred to PC, and then to AP, and then to photosystem 2. Based on the level of energies, it gradually can be transferred to the photosystem 2 for photosynthesis. So what's the benefit for that? If you look at these absorption spectrums of phycobility zones, you can see that it complements with chlorophylls. So we know that chlorophylls harvest light in the range of red light, and green and blue light. So that's why we can see plants as they look greenish, because they don't harvest green light. But with the help of phycobility zones, phycoerythrin, phycocyanin, allophycocyanin, they can harvest light in this range of green and yellow and orange. So that's why from the slides in the beginning, you'll see some cyanobacteria, they look brownish because they produce phycoerythrins and harvest the green portions of light. So they almost harvest all light in the visible spectrum. That's why they look pretty brownish or even black. So they can also change the composition of the rods when they are under different light conditions. So under green light, they can produce more of these phycoerythrins, so they can harvest green light. And under red light, they produce phycocyanin instead, so they can use red light.
So how fast they can how much time they need to make a change?
Several days. It's not a very fast process, because they need to remodel the whole phycobility zones and synthesize the new proteins and replace the rod's composition. So it's not a very fast process. Okay, any other questions so far? Okay. So let's keep learning what's cyanobacteria. So normally we use cyanobacteria as a model species to study photosynthesis. But why? Because cyanobacteria, in terms of photosynthesis, cyanobacteria and plants are pretty similar. So this is the photosynthesis happens in plants. You can see the photosynthesis divided into two parts: light-driven reactions and carbon reactions. And in the light reactions, the light energy is harvested by photosystem 2 and photosystem 1. And that reactions turns water to oxygens and store that light energy into chemical energy as NADPH and ATP. And that energy can be used for carbon benzene cycle in the synthesis of carbohydrates. So that's pretty similar in cyanobacteria and plants. And also the structure is also pretty similar but different. Because in cyanobacteria, that happens in a single cell. So a single cell looks like a chloroplast. But chloroplast is an organelle in plant cell. You can only find chlorophylls in chloroplast, but not the other parts of a plant cell. And also, they have different light harvesting complexes. So we know that in cyanobacteria, they use phycobility zone as an antenna complex so they can harvest more light. But in plants don't have phycobility zones. If plants have phycobility zone, when you look at the leaves, they will look pretty brown, but that's not true. So they have another antenna complex called light harvesting complexes. It's around photosynthesis 2 and photosynthesis 1 and help them to harvest more light energy. And so why do we study photosynthesis in cyanobacteria? Why don't we, also some people study that in plant directly, but what's the advantage for doing that in cyanobacteria? Because it's model organisms and photosynthesis is very similar to plants, and cell structure is simpler. It's a single cell, and smaller genome size, much more smaller than plants. For the plant, genome size could be several gigabase, but for cyanobacteria, it's several megabase. And easier genetic engineering so that we can easily do genetic knockout overexpressions, such kind of thing to study gene functions in cyanobacteria. And short life cycles. Although life cycle is longer than E. coli, they don't multiply every 30 minutes. Sometimes you need to wait 24 hours or even more for them to divide, but still much faster than plants. And that's corresponding to when can you get graduated. If you use cyanobacteria as a model, I guess you can graduate fast. And also, very important for us doing photosynthesis research is that cyanobacteria can grow photoautotrophically and also heterotrophically. So what does that mean? So photoautotrophically means that when you provide light to the organisms and it can grow. And heterotrophically is that you provide them carbon source like sugars or the other or acetate, that kind of carbon source that they can grow. So we know plant cannot do that. You cannot put the plants and then shut down the lights and put that in the sucrose medium and it grow up to a big tree. That's just not happens. But we can do that in cyanobacteria. We turn off the light and then we provide them sugars and they grow. So why is that important? Because we need to study the genes in the photosynthesis. Sometimes when you knock out the genes related to photosynthesis 2 or photosynthesis 1, the plant just die. Because if they don't do photosynthesis, they cannot grow. But if they can grow in the medians with sugar, and then we can knock out several genes of in the cyanobacteria and study their functions, they still grow and produce chlorophylls. So that's the reasons we like this organism very much in our study. So okay. Right, for some species of land, especially for the model organisms. Yeah. No, not all species. Yeah, we selected several of lands that can do that for doing experiments. Yeah. Okay, any other questions so far? Okay. Yeah.
Pretty naive question. So when you do research in plants, you cannot just knock out the gene of photosynthesis 1 or 2 specifically, right?
No.
Oh,
Yeah, when you
so that you cannot just knock out. Wait. Oh, you mean when you knock out some genes, because some genes are linked, so you and also knock out the photosynthesis gene, so that you cannot use plants to do some gene knockouts too.
Yeah. So there are many subunits in photosynthesis 1 and 2. Like in photosynthesis 1, there are 12 subunits, and photosynthesis 2 is 20 subunits. So it's a very, very large protein complexes. So sometimes when you knock out 1 or 2 of lengths in the complex, the complex falling apart. They don't assemble. They don't form a functional photosystem 1 or 2. And then if plants don't have functional photosystem 1 or 2, they just don't grow. They just die. Yeah.
So when you knock out the gene, you want to knock out non-photosynthesis related gene to do other research?
In cyanobacteria, we can knock out photosynthesis related genes. Because when they don't do photosynthesis, they can still uptake the sugars from the outside as their energy source. So in cyanobacteria, we can do that. Yeah, we can knock out genes related to photosynthesis. But in plants, it's difficult. Yeah. Okay. Any other questions so far?
So just curious. For example, we want to know how do the global warming affect the plants, the trees' photosynthesis. So can we use these bacteria to test what if the temperature increases, or what if the water limitation changes, or what if the CO2 concentration increases, or if change is a photosynthesis rate? Can we do this?
Yeah. So when the carbon dioxide concentration increase, that increase the photosynthetic rates in cyanobacteria. I guess that's predictable. And for the other kind of experiments, I think some people are doing that in using plants in a field experiments. Yeah.
So that's because, as you just mentioned, there are a lot of advantages of using this one. So from those studies using plants, they try to inject the CO2 concentration to double CO2. And then some of the studies they do find they try to close their stomach. Because right now the concentration of the CO2 increases. So they don't need to open that much of the stomach to conserve the water.
Right.
So I'm just curious whether this kind of behavior can be seen in this
So no stoma, but there's a specialized compartment. It's not an organelle, but it's a compartment in cyanobacteria called Carboxy Zone. So it can concentrate the CO2s inside that compartment, and so they can have a higher concentration of CO2 inside the cell. But if you grow these cells under a higher CO2 levels, they just don't produce these carboxy zones anymore. Because they might feel the carbon concentration is, the carbon dioxide concentration is high enough, so they don't produce these concentrating mechanisms. That's what happens in cyanobacteria. Okay. Okay, any other questions? Okay, so I just keep going. So talking about relationship between cyanobacteria and plants, so you can see that cyanobacteria evolved very early on the stage of on the Earth, and the green algae and land plants happens later. It's around 0.5 billion years ago. So cyanobacteria is basically an ancestor of algae and plants by endosymbiosis. So the first endosymbiosis produced red algae and green algae, and then there's another symbiosis called secondary symbiosis, and resulting in different other algae. And plants comes from green algae, the original plants around 470 million years ago. It's a different lineage of plants. So what's the other relationship between cyanobacteria and plants? So I said the cyanobacteria has a specialized cell called heterocyst that can do nitrogen fixations. And we know that when we grow plants, we want to provide them fertilizer. The fertilizer has fixed nitrogens, like the ammoniums or the nitrates. So if a cyanobacteria can fix nitrogens, that in some ways they probably can also produce fixed nitrogen for plants. And that happens in some plants. They do symbiosis with cyanobacteria and called Cyanobacteria cyanobionts. So in that case, cyanobacteria provide plants fixed nitrogens, and plants provide them fixed carbon in return. So, and that also changed the physiology of cyanobacteria. When the cyanobacteria have symbiosis with plants, they produce more heterocysts. They produce more heterocysts and specialize for nitrogen fixations. A very, very classic example is Alola, Ming-Yang Ho. You can also find it in 醉月湖 or 生态池, only campus. So Alola is a fern. It's a fern. And it has a central leaf cavity that is empty, and cyanobacteria are living in these leaf cavities. In that, they fix nitrogens and produce the nitrogen source for the plants. So it's a very classic symbiosis example for plants and cyanobacteria. And there are also other different examples in this, but I won't explain all of them today. So because cyanobacteria can fix nitrogens, so people can also use that as a source for fertilizer. So nowadays, people pay more attention on the organic farmings. They want to provide the fertilizers that are not chemical fertilizer, but the biological fertilizers. And cyanobacteria with a heterocyst could be a source for those fertilizers. But there's also other drawbacks for doing that. Because some cyanobacteria, not all cyanobacteria are good. For the good, I mean they are not for the good cyanobacteria, they are not toxic to humans. But cyanobacteria, some of them, they can produce compounds that are toxic to humans or animals or plants. So when you use that cyanobacteria, they produce toxins and provide that to the in the field for plants or crop that might kill your plants. And the mycocystin is a very famous toxin produced by some species of cyanobacteria. And so there's a serious problem for cyanobacteria when the water reservoir or the lake, freshwater lake, get contaminated by the cyanobacteria. It's called algal bloom, 藻华. So when it gets contaminated and produce toxins, it's not very easy to remove that from the water. So looking at this picture here, I guess you can make a guess what can cyanobacteria do to human beings, and how does cyanobacteria kill dogs? Toxin, right. So just don't if you see the water in the lake look like this, don't let your dog swim in the lake like this, and don't let them drink this kind of water. That might be toxic to your dog, and also to our human beings, for sure. Okay, so there's some application for cyanobacteria. As I mentioned earlier, now this is a very famous cyanobacteria that we can eat. It's called Spirulina, 螺旋藻. And the interesting thing here is that although they call it 蓝绿藻, 蓝藻, 螺旋藻, they are not, I want to express that again, they are not algae, they are not 藻, they are bacteria. So when you see people eating this 螺旋藻, you can tell them, do you know you are actually eating bacteria? Okay. Anyway, it's a very nutritious bacteria that can be used for nutrition supplements or used for feeding animals.
Is it because of nutrition or is it kind of probiotic?
It's because of nutrition, and because it's a very high protein content, and also it has many vitamins that are good for humans. So those cells they are died. They are died. Yeah. So
Curious. So this language is what I just learned from the elementary school. The first life in the earth. This language.
Yeah.
The same thing or different?
The same thing. Yeah, and also in the beginning of my talk, I said. Oh no, that's fine. The 蓝绿藻 might not be the first bacteria on the Earth, because the first bacteria get evolved. They don't do oxygenic photosynthesis, and because oxygenic photosynthesis happens earlier than the oxygenic photosynthesis, so. In the earlier stage of the Earth, the bacteria living there, they don't produce oxygen and they don't act oxygens. And the cyanobacteria is the one produce oxygen, so that must be happens later in the evolution of life. Yeah, it's a sensing.
Cyanobacteria can only do oxygenic photosynthesis. They cannot do non-oxygenic photosynthesis.
That's a good point. Actually, nowadays, some people find some special cyanobacteria they can do anosogenic photosynthesis, but they don't do this at the same time. They don't do anosogenic and oxygenic at the same time.
Machinery, but they don't do it at the same time. They cannot do it because it is oxygen and acid, so they can only. But they have two set of genes, so they can do both.
I think in that cyanobacteria, they lost the functions of oxygenic photosynthesis. Yeah. So, but that might happen at a later stage of the evolutions.
So that's more evolved
later.
More later cyanobacteria, not the first one. People don't have a very conclusive evidence right now, but because these bacteria, they can do, the horizontal gene transfer happens a lot in the bacteria. Sometimes they exchange the genetic components and then get the new functions, or they lose the functions. That's quite frequent, happen quite frequently in bacteria. So that they might gain it later, or it's evolutionally happen first. That we don't know. Yeah. Okay. So it's called 螺旋藻 because you can look at this. It looks like spiral shape, and people start eating that back to the 16th centuries in Mexico. And also there are other food about cyanobacteria you may have known, but you might not know they are cyanobacteria. They are 法菜 and 地木耳. So the 法菜 is just called hair vegetables, and 地木耳, 葛仙米藻, 雨来菇, and 地木耳. English names, very different English names. One interesting one is which is butter. 巫婆的奶油. So although these are all cyanobacteria, people call it vegetables, or call it's algae, mushroom, or 木耳 is also mushroom. So look at the common names, you don't know they are cyanobacteria, but they are actually cyanobacteria. And you can, I found this in Quanlian. In Quanlian, they mention it as 雨来菇, but it also said it's terrestrial cyanobacteria, cyanobacteria living on the land.
So these are cyanobacteria you may know and you may have eaten during your life. And what's the other application for cyanobacteria? So I have introduced you the phycobility proteins. That's the proteins that make the phycobility zones. They have different colors of lens, cyanide, and blue, dark blue, and pink. So what can we use for it? So you know it's very difficult to find the natural colors of blue. It's very difficult to find the natural color of blue. You can find the natural color of green. You can find the natural color of yellows and maybe red. But blue is very difficult. So in 2013, that's the first time that we got the natural colors of blue got approved by FDA in the US. That's applied by the companies that are making M&Ms. So now for the blue color, they also have natural colors for their chocolates now. So there are other applications in the industry, like the wastewater treatments. Because these cyanobacteria, they can uptake the nitrogens from nitrogen source from the water. So it can remove the nitrogen pollutions in the waters. And also, we can use cyanobacteria to produce biofuel. But these ideas was very innovated maybe back in 2013, maybe 10 years ago. But later, this kind of idea using algae or cyanobacteria to make biofuels faced some challenges. So I take this company as an example. It's algal companies in the US. Their goal at the beginning is to make ethanol, bioethanol as a fuel for biofuel by using cyanobacteria. So this company established in 2009 or 2008, and after 8 years, there's no commercial products got produced by Lens. So until 2015, they are still not in a commercial productions. So, and after that, they decided, okay, they changed their goal. They cut 25% of their staffs, and then changed their goal to water treatments and carbon capture now, and maybe fuels later. And in another year, 2016, it's 10 years of their anniversary, and they added algae-based sustainable products to its portfolio. So they basically changed their goal to produce bioethanol to algae-based sustainable products. And why? Why? So this is their main products right now. Yeah. So important thing for a company is that how to make more money. You can think, making this product make more money or making fuel. So look at the oil price right now. It's 27 NT dollars per liter for gasoline. How expensive is cosmetics? Maybe 100 milliliter and it's several thousand dollars. So it's a huge difference. So that's why, although many companies, their original goal is very good. They want to save the world and they want to produce sustainable biofuel, but at the end they tend to produce product that can make money. That's all.
At least it's from natural.
Yeah, it's from nature. Yeah. Okay. Okay.
Is that the real reason? The makeup as a alternative, is that the real reason making those companies to make those kind of algae-based sustainable product, or is it just because using algae to make biofuels is super inefficient, or it actually costs even more pollution?
That might be another reasons, yeah. I don't know the real reason behind that, but we can see the transformations of the directions of the companies. But people still making bioenergies in the US, but they still use basically crops. They use corn starch for making bioethanol. So maybe that's a little inefficient comparing to the traditional gasolines for the car engines, but truly making this kind of products, they can get more profits. Then I guess that's why, yeah, they transform to the other directions. Yeah. Okay, any other questions so far? So, okay, so what have we learned about cyanobacteria today? So first thing is that we don't call cyanobacteria as blue-green algae anymore. So they are bacteria and they are not algae. And cyanobacteria are important in the history of the Earth and the evolutions of life. Cyanobacteria are pretty like plants in terms of photosynthesis. And we can use that for academic research and also industrial productions. And we actually eat and use some cyanobacteria in our daily life. And you also learn how to not get your dog killed by cyanobacteria. And so, do you still want to learn more about how cyanobacteria use far light for photosynthesis?
Yeah.
After, okay. Okay, I'll take, yeah, maybe around 10 or more slides later. So, okay, so let's go to what I'm doing here about photosynthesis. So, photosynthesis, plants do photosynthesis using chlorophyll A and B. And looking at the spectrums, that's the energy from reach the surface of the Earth, you can see the chlorophyll A and B, they harvest light in the visible range. So the structure of chlorophyll A, B, DF are pretty similar. There's only one functional group's difference among modifications. But this modification of chlorophyll D and F, these two chlorophylls only found in cyanobacteria that allow them to harvest light in the range of infrared light. For the definition of infrared light, we call it the wavelengths between 700 nanometers to 800 nanometers. So in that way, they can harvest 35% of more energies comparing to the other organisms only use visible light. So that's beneficial for them to do photosynthesis in some area not limited to visible light. And the study of the oxygenic photosynthesis in infrared lights, it started around 20 years ago. So at least several important articles about that in the history of studying photosynthesis in infrared light. So you can see it's Nature and 8 years, another Science, 6 years, 4 years, 2 years, 2 years. So it's got extensively attentions in the recent decades that people are knowing that this is an important question and focusing on studying that. And including me, I'm doing the research related to these topics. So just give everyone a brief introduction for how cyanobacteria can utilize infrared light for photosynthesis. So for some of them, they can do a photoaccumulation process called infrared light photoaccumulations. That's a photoaccumulated process that can transform between visible light and infrared light. So under visible light, it just looks like other cyanobacteria. There's a phycobility zones, photosystem 2, photosystem 1, and chlorophyll A. So they use visible light for growth. But under infrared light enriched environments, they can sense the change of the light conditions, and then they produce chlorophyll D and F. and then remodeled their phycobility zones, photosystem 2 and photosystem 1. Some subunits in them are replaced, so they can bind to chlorophyll D or chlorophyll F. That allows them to harvest far light for photosynthesis. So it also controls, this process is controlled by a phytochrome-based regulators. So RPA-BC, RPA is a phytochrome. It can sense the change of the light conditions in red light or far light. So under far light, induce the downstream, it's activate downstream regulators. And this RPB is a transcription regulators that can regulate the genes in the phylic gene cluster, including lens self, chlorophyll F synthase, and possibly chlorophyll D synthase. It haven't been found yet. And PS1, PS2, and phycobility zone genes. So they can remodel the phycobility zones, photosystem 1 and 2, to allow them to harvest far light to growth. Okay. So these cyanobacteria can do that because they have a specialized gene cluster called phylic gene cluster. These clusters contain 20 genes, including the regulators, RP genes, and the subunits in photosystem 1, 2, and the phycobility zone to allow them to remodel these complexes to harvest far light. And you can see that this gene cluster has been found in different sections of cyanobacteria from 1 to 5. If you still remember that from 1 to 5, they have different morphologies, unicellular and filamentous cyanobacteria. They are all, these clusters are found in all of these sections. So we believe that it's horizontal gene transfer events between the cyanobacteria to allow the phylogenetics different cyanobacteria to having a closely related cluster. It's not being found in all cyanobacteria, only found in some of them. Nowadays, around 50 cyanobacteria has been found having this gene cluster. And there are around 1,500 of cyanobacteria has been sequenced. So only 3% of them has been found have this gene cluster. So the regulators will induce the genes expressions in the gene cluster under far light. So looking at this RNA sequencing results, you see this is a transcript level in white light. This is transcript levels in far light. And those genes that are highly induced have a higher transcript levels are the genes in the phylic gene cluster. It's very, very clean. You don't see other genes got induced such higher fold comparing to the genes in the phylic gene cluster. So the regulation is very specific and is highly controlled. And the level change is very high. You can see the level is around 10,000 difference between the levels in white light and far light. So what can we use that in the environmental studies? So this is not my research, but it's very interesting that because we know that the gene cluster only happens in the cyanobacteria that can utilize far light. And we can use that as a gene marker, like the gene APCE2. Use that as a marker to trying to find these genes in the environmental DNA samples. And what people found is that in the DNA samples that collected around the world, you can all find these genes, APCE2. So it is likely that this kind of cyanobacteria that can do phylic are widely distributed around the world. So if you have an environmental DNA, probably you can try to see if you can find these genes in your DNA, and that might tell you there are flower light using cyanobacteria in your sample. So in the environments, where can we exactly find those flower light using cyanobacteria? So talking about cyanobacteria, the first impressions is that they are living in the water, like the oceans or in a freshwater lake. But the flower light is actually not very penetrative into the water bodies. So you can see the water looks like blue. So because blue light have the best penetrations into the water bodies. And for the flower light, if you go deep to the oceans around 3 meters, anywhere lower than 3 meters, you don't see any flower light over there. But you still see blue light, like 100 meters, you still see blue light. So this is another key place that you will find flower light using cyanobacteria. But how about the terrestrial environments, like the shade of plants, sand? These kind of environments have higher ratios of flower light comparing to the visible light, because most of the visible light got absorbed by plants. So actually many of these organisms were isolated from terrestrial environments. And this is stromatolites. You can see this layer, this contains cyanobacteria inside that. And that's the first place chlorophyll F got isolated from the shark bait West Australia. And also, you see these microbial mats in the hot spring. It looks green. There might be cyanobacteria growing inside that. And also soil. Soil, you'll see this green color of cyanobacteria. This kind of soil crust, it's a symbiosis between, I remember it's fungi and cyanobacteria. And also, you also found these structures at Miaoli Huoyan Shan. This bacteria was isolated by Dr. Wu Junzong. This is microbial mats in hot spring. And you do the sections of that, you see only the top layer is green. So light cannot penetrate too much. This very dense layer of microbes that harvest almost all the lights over there. And you will see at the lower layer, they can probably only use flower light for growth. And then, yeah, I mentioned algal blooms. The visible light is strongly filtered by algae. So in that environment, might be flower lights also enriched for these cyanobacteria to grow. So these are some environments, places that we can find those flower light using cyanobacteria. And this is my favorite pictures showing where can you find flower light using cyanobacteria in the forest. So you can see the full spectrum of light coming from the sun. And then the plant leaf, they filter the visible light. And only the flower lights can well penetrate under the canopies. And maybe over there on a stone, you can find in this shaded cyanobacteria that can utilize flower light to grow. That's probably the ecological importance of these flower light cyanobacteria, because they can grow on the places that we didn't believe that cyanobacteria can grow in the past. And that might change people's views on primary productions all over the earth. So, but basically my approaches and my research is more about the molecular biology and the physiologies of these cyanobacteria that can utilize flower light. But also I'm trying to do some sampling and analysis of the metagenomic data to see where can we exactly find those flower light using cyanobacteria in the environments, and how abundant what's the abundance and the distributions of land corresponding to the abundance of far red light. So in my lab, mostly I do traditional microbiology works and molecular biology, genetics, organic chemistry, and spectroscopy. So here I would like to introduce some special techniques we use for studying cyanobacteria. So first one is absorption spectroscopy. So these cyanobacteria, when they grow under different light environments, they show different colors. But only by using the absorption spectra, we can tell how different they are. So they have absorbed different spectrum of light, and only the cells in the far red light, they can absorb far red light beyond 700 nanometer. And we also use fluorescent spectroscopy, including the room temperature and the low temperatures fluorescent spectroscopy. So how is that useful? So for the low temperature spectroscopy, you can see differentiated fluorescence coming out from photosystem 2 or photosystem 1. So these signals are from photosystem 2 and these from photosystem 1. And that signal is different from when they are under different light conditions, and also are different from species to species. But using the room temperature fluorescence, you only see a block fluorescence coming out from chlorophyll A, and this one is from chlorophyll F. So with these two skills complement with each other, we can know more about the compositions of the pigments in the cells and how are they binding to different photosynthetic complexes, and how does that change the fluorescence properties of them. And we also use HPLC to quantify and differentiate different pigments like chlorophyll A, F, and D. Although they are quite similar in their structures, we can still separate them using the programs running in our lab. So another way we can do also establish the way that we can do genetics in these cyanobacteria. Because for those cyanobacteria, they doing FALIP, they are non-model organisms. So they were not genetics system available, allow you to do the overexpressions or genetic knockout. And this is a systems I developed during the times when I was in the US. I developed the knockout systems for these FALIP cyanobacteria. And now here's my students. He successfully established the way to do overexpressions in these two organisms. So for this organism, we can now have a full genetic system. We can study the gene functions in them. And how does that technique useful? So we can provide you the basic knowledge for cyanobacteria based on spectroscopies, and you can tell the function of the photosynthesis. So this is a recent collaborations we have with Dr. Li Feiwei at Cornell University. So we use this spectroscopy to analyze the pigment compositions and the property of the photosystem 2 and photosystem 1 and in these specialized cyanobacteria. This is a very early evolved cyanobacteria that doesn't have thylakoid membranes. So as I mentioned, cyanobacteria has a thylakoid membranes and the photosystem 2 and 1 sitting on that. But this one doesn't have thylakoid membranes, and all the photosystem 2 and 1 are sitting on the plasma plasma membranes, right? So that's characterizing this organism can tell us more about the evolutions of photosynthesis and cyanobacteria. So my current research topic at NTU is still focusing more about the cyanobacteria using far red light to characterize the diversity distributions, the mechanisms, and try to use the regulatory tool to control gene expressions in far lights. So just talking in brief, then we try to, at the beginning, we try to collect samples from the NTU campus and try to isolate the bacteria or algae that can utilize far light for photosynthesis. What we did was basically collect the samples and put them to the medians that cyanobacteria like to grow, and then only use far light shining on the samples. So in that way, we can select the cyanobacteria that can use far light to grow. And so far, we found 2 cyanobacterial strains and 5 algal strains that can grow in far light. And now we also collaborate with Dr. Xiao Youjing from NTU to trying to collect more samples to characterize and isolate more strains. So the other interesting topic, as I mentioned about the RNA sequencing data of far light, that these genes in the far gene cluster, they got highly induced under far light, and the difference can up to 10,000 folds. So it's quite a huge difference and the tight controls. So we're trying to make that as a synthetic promoters by fuse that with reporter genes, YFP. So if we can do that, and we can control the gene expressions by providing different light spectrums or different intensity of light to control gene expression, that would be pretty useful for in the view of synthetic biology. But to do that, we need to first establish a conjugative system to doing that. That's an overexpression system. So what we did was using the constitutive promoter first, and then use that to express the YRP genes. And use that to use conjugation to deliver that plasmids into cyanobacteria. And currently, the result was successful so far that now we can do expression of genes in the 2 non-model cyanobacteria that we selected. This one is the model cyanobacteria that people already know that we can do overexpressions and genetics in that. But for these 2 strains, we are the first one to use this technique to transfer genes inside these strains. So we are now trying to test other far light inducible promoters in these strains. So, and hopefully there will be more capacity for us to do genetics in those strains. So overall, that's most about my research. I'd like to thank my lab members that they help a lot for me to establish the techniques and also the infrastructures in the lab, so we can finish setting everything in a short time and start doing our experiments. And thanks to the other collaborators, and also thank you for coming to the talk today. If you have any questions, I will be very happy to discuss any of that with you. Thank you. With other organisms. That's probably because of the way that we use to isolate those strains. Because in the methods people using to isolate those strains, they collect the samples and put them into the median of cyanobacteria. and then see if they can grow under flower light. But for the symbiontant cyanobacteria, sometimes they lose the function to grow by themselves. So if you're using the selection method like that, you sometimes don't find any symbiontant cyanobacteria can be isolated. So yeah, the short answer is that we don't know that so far. Yeah.
Is this group phylogenetically conserved?
For the gene cluster?
Yeah, for that, the strain that has far red light capacity, are phylogenetically conserved?
Not really. Because, so for the gene cluster they have a second set of photosynthesis 2 and photosynthesis 1 subunits. And the genes in the gene cluster are phylogenetically conserved between different cyanobacteria, only for the gene cluster, but not the cyanobacteria themselves.
Okay. So you cannot use like cysteines or something to decide whether that takes up the capacity of far red light?
That might be difficult to do that directly, but I'm trying to collect all the 16S sequence of the cyanobacteria that can do far leap, and trying to make a small database of all the organisms can do that, and to match that to the database to see how much sequence I can find. But that might not be a direct evidence until we can see those genes in the gene cluster in the species. Yeah.
But it's possible to use some environmental DNA method to identify the resistance.
Right. It's
target, not cysteine, some target specific gene.
Yeah, some specific gene that might be useful.
Can you go back to the slide, the forest and the sunlight and the far red light?
This one?
So if in terms of, so I'm not sure how much of the far red light can penetrate it to be absorbed by this cyanobacteria, like in terms of percentage or
Yeah, here is another figure that's showing, you can see the panels in the middle. So the solid line is the energy from the sunlight. And the dashed line is the energy that penetrate the canopy. Penetrate the forest top canopy. Penetrate the forest canopy. So almost all the far lights can penetrate the canopy. Plants just don't absorb that.
those bacteria can absorb it.
Yeah, those bacteria can utilize that.
And then they can do, so when they do the photosynthesis, they release H2 also.
They release oxygen.
Oxygen.
That is oxygen.
So will they release water as well?
They
open their stomach, or they don't have this kind of
they don't have stoma. They release oxygen. Yeah. Yeah, they take carbon dioxide, right. 对，它就是直接跟外界 exchange 那个空气这样子 ，exchange 直接 exchange。对。
Excuse me, can we go back to the previous slide? So, the forest.
The forest one. Oh, you are interested about the forest.
Really like forest.
Yeah, we are more like outdoor experimenter. So is there a way to measure this outside?
The light spectrum?
No,
you mean?
the photosynthesis of all these cyanobacteria outside.
For the particle rates?
In the field?
Yeah.
Rates?
Yeah.
In the field?
The reason we say that is we are doing that for the sun-induced fluorescence. And I think the major challenge is because sunlight and the radiation, they are the same unit. But the thing is, solar radiation is always much stronger. All the photosynthesis, the energy that induced by sunlight. So there's only very narrow bands that you have. I don't know how to pronounce that. Frontal line, but it's just only certain very narrow bands. Got a weaker solar radiation. So, because the reason I come here probably because we are measuring the canopy.
Okay.
But the thing is, so I saw the red light. So first, I was like, actually, I come here to listen to the last 10 slides. Now, how you do it, but what I see is actually you measure in the lab.
Yeah, we mostly do that in the lab.
But is there, because that would be a very good collaboration, because if there's a way we would be able to assess how much carbon uptake at the stem level. So that would be like really
Important step.
yeah, because and many
okay.
like or ocean.
I have thought.
I was talking about journal.
Using carbon.
Yeah, that's why I say if we can measure this part outside, that would be really, because I don't know what's the peak of the chlorophyll F. Is the peak absorbance is what wavelength?
It's around 707, 705, 705.
Yeah, that's okay. Because I'm not quite sure is that related to solar radiation. I'm not quite sure because I'm more doing the satellite global optical science. So I was like really curious about how if there's a way to measure this at least at the same scale. So that'll be very interesting.
If you want to see if they are doing carbon fixations and the rate of carbon fixation, I guess it's pretty hard. It's pretty hard. For the methods I know, I don't know if there is a good way to do that outside. But if you just want to tell if they are chlorophyll F over there, that might be easier. We can use fluorescence to do that. But still, we need to collect the samples and then take that from, for example, take that from the rocks, and we can use a spectroscopy, fluorescent spectroscopy, to see if doing that in field is I don't know if there's an instrument can do that.
Do it because it's hard.
Yeah, we do it because it's
Very true.
Yeah, it's important questions indeed. Yeah.
hard to measure, easy to publish.
It will be important findings, I am
that's
sure.
very
Yeah.
important. Because I think it would be really nice we measure this and simultaneously doing, in a lot of ways we have frost tower, so you know the amount of carbon sequestration of the whole system. And then people doing regular canopy photosynthesis. But if that, there could be a gap between the measure from the frost tower and the actual measurement.
Right.
So there could be some of them from all this kind of, but they could be substantial. We just don't know that much.
Yeah, maybe. Yeah.
Yeah.
So what you were saying was that maybe these organisms living on the ground, they may contribute to the whole system for carbon fixation.
Yeah. 35%. No,
Wow, you find that's nature, okay? You find that's
come on, Marcus. No, I say that is 35% more in terms of.
Yeah, that will be enough.
no, 5% would be nature. If you find that's 5%, then you have it.
It's amazing. So,
It's a myth thing. So,
Yeah.
I think we can talk more about.
Yeah,
nature.
definitely, yeah.
Yeah, because we have like a very narrow band spectroscopy. Very, very narrow, it's a 0.8 nanometer. I don't know how, so we can really capture like a very tiny change from the red to far red.
Okay.
But the thing is, we always have challenge because of solar radiation. You gotta have sunlight to have photosynthesis. But that's also that's why they make it so hard to tell the difference between the energy from the plant and the energy from the solar radiation.
Right.
The same unit. But in the lab, because you can get rid of that part.
Right.
Here, outside is just hard. So anyway.
Yeah, maybe you can discuss more, and I can learn more about the techniques you use, and probably there's some possibility for. Oh, okay.
She's working on that one.
Question? No? Okay, no, I think Mia will still be here for a few minutes, and you still hungry, there's food outside, so let's give thanks to Mia, very nice talk.